Chapter 3 Plant Kingdom. In the previous chapter, we looked at the broad classification of the living organisms under the system proposed by Whitaker 1969, wherein he suggested the five kingdom classification via the Monera, Protista, Fungi, Animalia, and the Plantae. In this chapter, we will deal in detail with the further classification within the kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and the members of the Monera and the Protista having cell walls have now been excluded from the Plantae, though earlier classifications placed them in the same kingdom. So, the cyanobacteria that are also referred to as the blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms and the plantae. Let us also look at the classification within the angiosperms to understand some of the concerns that influence the classification system. The earlier systems of classification used only gross superficial morphological characters such as the habit, color, number, and shape of the leaves, etc. They were based mainly on the vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on few characteristics. Also, the artificial systems gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by the environment. As against this, natural classification systems developed which were based on natural affinities among the organisms and consider not only the external features but also the internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for the flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, the phylogenetic classification systems based on the evolutionary relationship between the various organisms are acceptable. This assumes that the organisms belonging to the same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. In numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like the chromosome number, structure, behavior, and chemotaxonomy that uses the chemical constituents of the plants to resolve the confusions, are also used by taxonomists these days. Algae. Algae are the chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of the other habitats like the moist stones, soils, and wood. Some of them also occur in association with the fungi, lichen, and animals, example the sloth on the sloth bear. The form and size of the algae is highly variable, ranging from the colonial forms like the volvox and the filamentous forms like Eulothrix and Spirogyra. A few of the marine forms such as the kelps form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of the two gametes dissimilar in size as in species of Eudorina is anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. Example is the volvox and the fucus. They are useful to man in a variety of ways. At least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of the dissolved oxygen in their immediate environment. They are of paramount importance as the primary producers of energy-rich compounds which form the basis of the food cycles of all the aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of the marine algae used as food. Certain marine brown and red algae produce large amounts of the hydrocolloids, water-holding substances. Example, algae, brown algae, and keratin, the red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular algae rich in proteins, is used as food supplements even by space travelers. The algae are divided into three main classes: the Chlorophyceae, Phaeophyceae, and the Rhodophyceae. Chlorophyceae. The members of the Chlorophyceae are commonly called the green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of the pigments chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contains proteins besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made up of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by the formation of the different types of the spores. Asexual reproduction is by flagellated zoospores produced in the zoosporangia. The sexual reproduction shows considerable variation in the type and the formation of the sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Pyrogyra, and Chara. Phaeophyceae. The members of the Phaeophyceae or the brown algae are found primarily in the marine habitats. They show great variation in the size and the form. They range from simple branched filamentous forms like ectocarpus to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of the xanthophyll pigment, fucoxanthin, present in them. Food is stored as complex carbohydrates, which may be in the form of laminaria or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and the nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe, and a leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of the gametes may take place in the water or within the oogonium, oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Rhodophyceae. The members of the Rhodophyceae are commonly called the red algae because of the predominance of the red pigment R phycoerythrin in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of the water and also at depths in the ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Asexual reproduction is oogamous and accompanied by complex post-fertilization developments. Common members are Polysiphonia, Porphyra, Gracilaria, and Gelidium. Bryophytes. Bryophytes includes the various mosses and liverworts that are found commonly growing in the moist shaded areas in the hills. Bryophytes are also called as amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in the plant succession on the bare rocks or the soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by the unicellular or the multicellular rhizoids. They lack true roots, stems, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence it is called as gametophyte. The sex organs in the bryophytes are multicellular. The male sex organ is called the antheridium. They produce the biflagellate antherozoids. The female sex organ is called the archegonium, is flask-shaped and produces a single egg. The antherozoids are released into the water where they come in contact with the archegonium. An antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called the sporophyte. The sporophyte is not free-living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce the haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for the herbaceous mammals, birds, and other animals. Species of the sphagnum, a moss, provide peat that have long been used as fuel and as a packaging material for the transshipment of the living material because of their capacity to hold water. Mosses, along with lichens, are the first organisms to colonize the rocks and hence are of great ecological importance. They decompose the rocks, making the substrate suitable for the growth of the higher plants. Since mosses form dense mats on the soil, they reduce the impact of the falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as the banks of the streams, marshy ground, damp soil, barks of the trees, and deep in the woods. The plant body of a liverwort is thalloid. Example, Marchantia. The thallus is dorsiventral and closely pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of the thalli or by the formation of the specialized structures called gemma, singular gemma. Gemmae are green multicellular and asexual buds which develop in small receptacles called the gemma cups located on the thalli. The gemmae become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on the different thalli. The sporophyte is differentiated into food, seta, and capsule. After meiosis, the spores are produced within the capsule. These spores germinate to form the free-living gametophytes. The predominant stage of the life cycle of a moss is the gametophyte, which consists of the two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping green, branched, and frequently filamentous stage. The second is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through the multicellular and the branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into the sporophyte consisting of food, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophytes. The pteridophytes includes the horsetails and the ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionary, they are the first terrestrial plants to possess vascular tissues, the xylem and the phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, and shady places, though some may flourish well in the sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissues. The leaves in the pteridophytes are small microphyls or as in Selaginella, or large macrophyls as in ferns. The sporophytes bear a sporangia that are subtended by the leaf-like appendages called the sporophylls. In some cases, sporophyll may form distinct compact structures called the stobili or cones like Selaginella equisetum. The sporangia produce spores by meiosis in the spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called the prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted environment requirement and the need for water for fertilization, the spread of the living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and the female sex organs called antheridia and archegonia respectively. Water is required for the transfer of the antherozoids. The male gametes released from the antheridia to the mouth of the archegonium. Fusion of the male gametes with the egg present in the archegonium results in the formation of the zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro, large, and micro, small spores, are known as heterosporous. The megaspores and the microspores germinate and give rise to the female and the male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes into the young embryos takes place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: the Psilopsida, Psilotum, Lycopsida, Selaginella, Lycopodium, Synopsida, Equisetum, and Pteropsida, the Dryopteris, Pteris, and Ediantum. Gymnosperms. The gymnosperms. Gymnos means Neet and sperma means seeds, are the plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include the medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally taproots. Roots in some genera have fungal association in the form of mycorrhiza pinus, while in some others, like cycas, have specialized roots called coralloid roots are associated with the nitrogen-fixing cyanobacteria. The stems are unbranched cycas or branched as in pinus and cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce their surface area. Their thick cuticle and the sunken stomata also helps to reduce water loss. Gymnosperms are heterosporous. They produce haploid microspores and the megaspores. The two kinds of the spores are produced within the sporangia that are borne on the sporophylls, which are arranged spirally along an axis to form the lax or the compact stobili or cones. The stobili bearing the microsporophylls and the microsporangia are called the microsporangiate or the male strobili. The microspores develop into the female gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of the pollen grain takes place within the microsporangia. megasporangia. The cones bearing the megasporophylls with the ovules or megasporangia are called the macrosporangiate or the female strobili. The male or the female cones or the strobili may be born on the same tree, Pinus. However, in cycas, male cones and the megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by the envelopes and the composite structure is called an ovule. The ovules are born on the megasporophylls which may be clustered to form the female cones. The megaspore mother cells divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develop into the multicellular female gametophyte that bears 2 or more archegonia or the female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and the female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophyte. The pollen grains are released from the microsporangia. They are carried in the air currents and come in contact with the opening of the ovules born on the megasporophylls. The pollen tube carrying the male gametes grows towards the archegonia in the ovules and discharges their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and ovules into seeds. These seeds are not covered. Angiosperms. Unlike gymnosperm where the ovules are naked, in the angiosperms or the flowering plants, the pollen grains and the ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. They range in size from the smallest Wolfia to the tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. The dicotyledons are characterized by seeds having two cotyledons, reticulate venation in the leaves, and tetramerous or pentamerous flowers, that is, having 4 or 5 members in each floral walls. The monocotyledons, on the other hand, are characterized by single cotyledonous seeds, parallel venation in the leaves, and trimerous flowers having 3 members in each floral walls. The male sex organ in a flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anthers, the pollen mother cell divide by meiosis to produce a microspores which matures into the pollen grains. The female sex organ in a flower is the pistil. Pistil consists of a swollen ovary at its base, a long slender style, and the stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has 3 celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cells, and 2 polar nuclei. The polar nuclei eventually fuse to produce the diploid secondary nucleus. Pollen grain, after dispersal from the anthers, are carried by the wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grows through the tissues of the stigma and the style and reach the ovule. The pollen tubes enter the embryo sac where the 2 male gametes are discharged. One of the male gamete fuses with the egg cell syngamy to form a zygote. The other male gamete fuses with the diploid secondary nucleus to produce a triploid primary endosperm nucleus pen. Because of the occurrence of 2 fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons, and the primary endosperm nucleus develops into the endosperm, which provides nourishment to the developing embryo. The synergids and the antipodals degenerate after fertilization. During these events, the ovules develop into the seeds and the ovaries develop into the fruits. The life cycle of an angiosperm is shown in figure 3.6. Plant life cycles and alternation of generations. In plants, both haploid and the diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, the haploid and the diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of the generation between the gamete producing haploid gametophyte and the spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following structures. Patterns. 1. Sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygotes results in the formation of the haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, pyrogyra, and some species of the Chlamydomonas represent this pattern. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few-celled haploid gametophyte. This kind of life cycle is termed as haplontic. An algae fucus species represents this pattern. In addition, all the seeds-bearing plants, that is, the gymnosperms and the angiosperms, follow this pattern with some variations wherein the gametophytic phase is few to multi-celled. 3. Bryophytes and pteridophytes interestingly exhibit an intermediate condition called the haplodiplontic. Both phases are multicellular, however, they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alternates with a short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All the bryophytes represent this pattern. The diploid sporophyte is represented by dominant independent photosynthetic vascular plant body. It alternates with the multicellular saprophytic or autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as the haplodiplontic life cycle. All the pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them such as the Ectocarpus, Polysiphonia, kelps are haplodiplontic. Fucus, an algae, is diplontic. Summary. Plant kingdom includes the algae, bryophytes, pteridophytes, gymnosperms and the angiosperms. Algae are the chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic organisms. Depending on the type of the pigments persist and the type of the stored food, algae are classified into 3 classes, namely the chlorophyceae, phaeophyceae, and the rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by the formation of different types of spores, and sexually by the formation of gametes which may show isogamy, anisogamy, or oogamy. Bryophytes are the plants which can live in soil but are dependent on the water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by the rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of the liverworts is thalloid and dorsiventral, whereas mosses have a upright, slender axis bearing spirally arranged leaves. The main plant body of the bryophyte is gamete-producing and is called a gametophyte. It bears the male sex organs called the antheridia and the female sex organs called the archegonia. The male and the female gametes produced fuse to form a zygote which produces the multicellular body called the sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant body is a sporophyte which is differentiated into true root, stem and leaves. These organs possess well-differentiated vascular tissues. The sporophytes bear sporangia which produce spores. The spores germinate to form the gametophytes which require cool, damp places to grow. The gametophytes bear the male and the female sex organs called the antheridia and the archegonia respectively. Water is required for the transfer of the male gametes to the archegonium, where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which the ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed and therefore these plants are called Neet, the seeded plants. The gymnosperms produce microspores and megaspores which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls and megasporophylls are arranged spirally on an axis to form the male and the female cones, respectively. The pollen grain germinates and the pollen tube releases the male gametes into the ovule where it fuses with the egg cell in the archegonia. Following fertilization, the zygote develops into embryo and ovules into the seeds. In angiosperms, the male sex organ stamen and the female sex organ pistil are born in a flower. Each stamen consists of a filament and an anther. The anther produces the pollen grains male gametophyte after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or the embryo sac which contains the egg cell. The pollen tubes enter the embryo sac where the two male gametes are discharged. One male gamete fuses with the egg cells in gamete and the other fuses with the diploid secondary nucleus, triple fusion. This phenomenon of tube fusion is called the double fertilization and is unique to angiosperms. The angiosperms are divided into 2 classes, the dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plants, there is alternation of the generation between the gamete-producing haploid gametophyte and the spore-producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycles, haplontic, diplontic or intermediate. Thank you.